Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 11, Clinical Microbiology, Laboratory, Diagnosing Infectious Diseases. So in this lecture, we will give you a brief overview of our clinical labs, talk about sample collection and handling, microscopy and microbiology, mention some culture methods, and continue down to discuss some challenges and end with a summary. Now, what do we do in the lab? So the purpose is to identify infectious agents, bacteria, viruses, fungi, parasites. Furthermore, to guide treatment by determining antimicrobial susceptibility, to support infection control and public health efforts. Key activities: processing patient samples like blood, urine, swabs, and conducting tests for pathogens. So microbiology labs act like detectives, figuring out what germs are causing an infection and helping doctors choose the right medicine to fight those. Now, sample collection and handling. So common sample types include blood, urine, stool samples, sputum, and wound swabs. Best practices: proper collection techniques to avoid contamination. Timely transport to lab to preserve sample quality. Challenges: contaminated or improperly stored samples give inaccurate results. Now, some types of microscopy used in microbiology. Well, we have light microscopy, so stains like gram stain for bacteria. Fluorescence microscopy highlights specific microbes under fluorescent dyes. Electron microscopy, high detail for viruses and ultrastructures. So microscopy provides a quick preliminary look at pathogens. Culture methods. So the idea is to grow microbes from patient samples to identify them. Types of media used: enriched media. It supports fastidious bacteria. Example, blood agar. Then we have selective media, where it inhibits some microbes while promoting others. Then we have differential media, helps differentiate microbes based on its appearance, like its color change. Clinical relevance. So culture confirms the presence of pathogens. So culturing is like growing plants in a garden. Different soils, quote unquote media, help grow specific microbes, and once they grow, we can identify them. Molecular diagnostic techniques. So methods include polymerase chain reaction. This helps us detect microbial DNA or RNA. We have next generation sequencing, provides detailed genetic analysis. The advantages here are that are rapid and highly sensitive and useful for hard to culture microbes like viruses. Serological tests, of course, looking at the blood, helps us detect antibodies or antigens in a patient's blood. ELISA test helps detect antibodies to infections like HIV. Rapid antigen tests help identify infections like strep throat or COVID-19. So it's useful for diagnosing current or past infections. Because indeed, if we find that we have antibodies for something, it means we've probably been infected before by that thing. Antimicrobial susceptibility testing. So the purpose is to determine which antibiotics are effective against a bacterial infection. Methods include Kirby-Bauer test or disc diffusion, broth dilution for minimum inhibitory concentration, MIC. So this guides doctors in choosing the right antibiotic. Now, quality control in microbiology labs. So the importance is to ensure accuracy and reliability of test results. The processes are regular calibration for equipment, running control samples alongside patient tests. Challenges include maintaining high standards with limited resources. So quality control ensures lab tests are accurate, like double checking every step in a recipe to make sure the final dish is perfect. Don't you just love these metaphors? And then some challenges in clinical microbiology include antimicrobial resistance and limited access to advanced diagnostic tools in low-resource settings. We can resolve a lot of these through global collaboration and investment in new technologies. So in summary then, clinical microbiology labs are essential for diagnosing and managing infectious diseases. Advanced tools like polymerase chain reaction and AST improve diagnosis and treatment. Collaboration and innovation are critical to address future challenges. And that's the end of this lecture. Continue now to Chapter 12.